Hi, am I in the picture? Okay. Yes? Hi, I'm Miss Sloan, and this wonderful group of students right here that I love are my honors bio students, and we are reviewing for Unit 5 for us, and that's all about heredity. Let me make myself smash that. So I broke it down on what you needed to know for each chapter. So it's like I'll have what you need to know, and then I'll have the review slides, and then what you need to know, just so you know. Okay, Nikki, we don't know why the lights aren't going on that. No, they were off. Sorry, that's a fish tank issue. So for the first one about chapter 5, I want to remind you, don't forget to do your additional objectives, right? Because you could have a question over your additional objectives. Look through that. I don't think I made it so you could write on the slide, and I apologize. I totally forgot to do that. Can you write on the slide? I'm sorry, my bad. So let's read through it. Diagram the cell cycle, name the parts, what occurs in each, and identify the controls of it. Can you diagram a cell in the stages of mitosis with a cell that has 3 chromosomes, 4 chromosomes, 5 chromosomes? Can you name and describe 3 different ways meiosis contributes to genetic variety? Can you describe the similarities and differences between a haploid and a diploid cell, mitosis and meiosis, meiosis 1, meiosis 2, cytokinesis in plants and animals, oogenesis and spermatogenesis? Since you can't highlight right now, check in with your bio buddy. Which ones are most concerning to you of that? Don't say all of it because you know you're good at some things in there. Okay, go over that. Okay, now come back to me. If there's anything there that I don't hit today or you feel like I didn't help you enough with that, then when we're all done, ask me those as your questions, okay? Make comments on that and ask me those questions. So the first thing I want to go over is the cell cycle. Let's go, Pear Duck. Let me try again. Come on, Pear Duck. Go. Is it going? There we go. So on your Pear Deck, and I just posted an old TikTok just a few minutes ago on that. If you are a TikToker, but what we want to remember is G1, right? What happens during G1? The cell grows and you make more, and that makes sense to us. We don't need to memorize that because we know we just completed what? Mitosis and cytokinesis. And so it would be a really good idea for us to have that cell start to get bigger and make more organelles. In the S stage, that's when DNA does DNA replication. This is when you form the sister chromatids. This is when you are doing your modeling of your cell. Thanks, Nikki. And I can't open up with this bag, sorry. This is when you were doing the modeling of your cell. Let's say you had 2 chromosomes only in this cell. If this black one came from dad, this one would have to come from mom, and this would be a homologous pair, right? Same size, same shape, and they code for the same characteristics. Then each of them, DNA would replicate, and you still have a homologous pair, right? But each member of the pair consists of sister what? Chromatids. And in mitosis, the whole thing about mitosis is you copy all the chromosomes. So we copied this chromosome and we copied this chromosome, right? And in mitosis, the whole point is to line them all up, then make sure we line them up in the middle, and then we separate the sister chromatids from each other so that each daughter cell would get a copy of the big black, the big red, and this one we get a big black, big red. You okay with that? But when we do meiosis, we still have our homologous pair, right? In its S stage, you're still going to replicate, but this time we're going to go through all the stages, not once, but twice, okay? And the first time, prophase 1, the chromosomes become visible, and then what can happen? Synapses. And this is where crossing over could occur. So I could break off a little licorice here, right? And so I'd have a little red right here and a little bit black right here. That's crossing over. That is prophase 1. Then you have metaphase 1. They meet in the middle at metaphase 1. Can I do independent assortment with just 2 chromosomes? No, I cannot. I would need another set of homologous pair, and then it could line up any which way it wanted. What is that called? Independent assortment. Then we do away at anaphase 1, but this time we separate what? The homologous pairs. And then telophase 1 cytokinesis, and how many cells do I have? 2. They are both considered haploid because I don't have the parent chromosomes together anymore. Mom and dad chromosomes went through a divorce, right? So if the divorce happens, right, then you are a haploid cell. Now we're going to go prophase 2. Can we do any crossing over? No, we have nobody to cross over with. Our pair has been separated. We do metaphase 2. Can we do independent assortment? Even if I had another set down here, could I do independent assortment? No, because I don't have homologous pairs. They're not, it doesn't make any difference at that point. And then we go away at anaphase, and now we're going to separate the sister chromatids. Okay, turn towards your bio buddy. See if they get the difference between mitosis and meiosis. Check with them. Okay, come back to me again. Mitosis, I copy them all, I line them up single file, and I separate them. When I'm doing meiosis, which is getting ready for sex, I copy them all, but they line up with their homologous pair. And meiosis 1, I separate the homologous pairs. Meiosis 2, I separate the sister chromatids. 2 divisions. Okay? So this making of the sisters is what occurs during the S stage of interphase. That's when you make the sister chromatids. Then G2, I'm making proteins that I might need for this process. And then M just is all about the nucleus. It's the chromosomal dance. Whether you're doing mitosis or meiosis, it's your chromosomal dance. Then everything else, we call that cytokinesis, division of the cytoplasm. Yes. Sure. If I had, let me get some extra here. Let's say I had a cell with 4 chromosomes in it. Am I in the picture? Okay. So if I have a cell with 4 chromosomes in it, 1, 2, 3, 4. Let's say these black ones came from dad, these red ones came from mom. During this stage right here, the S stage of interphase, this one is going to replicate, this one is replicating, this one is replicating, and need more hands, and this one is replicating. These are sister chromatids held together by a centromere. This is the homologous pair right here. That's a homologous pair. And this is a homologous pair. Is that it? Yeah. Okay. Anybody else on that? I know for you, the licorice modeling was a really hard thing, but it forced you to see if you really understand it. It was a thinking activity to see if you could process that information. And I could catch where you had misconceptions by correcting you on that. Okay. All right. So then it is either going to be the M is either mitosis or meiosis, but cytokinesis is going to happen where you divide everything else. Right? Now, when we look at the controls of that, okay, remember the analogy I gave you when you go to the airport? You have to have 2 things. You have to do 2 things. You have to have a what? Ticket, and you have to clear security. The ticket is called cyclins. There are multiple different cyclins to control the cell cycle. There's M cyclin, S cyclin. There's so many different, I don't have you memorize all of the cyclins. Okay. Security is wherever you see one of these stop signs. This is a really important one right here because you've just finished with mitosis and we want to make sure you did it right. Okay. Or we've replicated the DNA. Did you do it correctly? We're going to check it right here. Right here in the middle of mitosis, remember we talked about what happens during any kind of metaphase. They are what? They're either meeting in the middle at metaphase 1 or meet in the middle, right? They're meeting in the middle. So you have a checkpoint right there where you're like, pause. Everybody in the middle? Everybody lined up? We good? So you check right there. Those are checkpoints. That's like going through security. being able to have the right chemicals and cell signals to move on to the next step, those are cyclins. That's how the cell cycle is controlled. And we're going to be talking about in our, oh, we talked a little bit. We talked about cancer, right? When the cell is out of control. Yeah, okay. All right, so then programmed cell death, they'll try to fix it, but if you can't fix it, then it's apoptosis. All right. Then the next two slides are just about the stages of the cell cycle. So why don't we go ahead and we will do mitosis together, okay? So here, am I in? Okay, so let's do it. Okay, so the whole thing's called mitosis. The first stage is called prophase. The chromosomes become visible. I want to remind you, how many chromosomes are in this cell? 2. Where's the homologous pair? Right here. What are these? Sister, what are these? Good. Okay. The chromosomes become visible. The nuclear envelope and the nucleolus disappears, radiating microtubules, asters. They meet in the middle at metaphase, away at anaphase. Who's going away? Sister chromatids. Okay. Away at anaphase to nuclei at telophase, cytokinesis. Okay, now because I'm big and in the middle, let's just do meiosis right now, and then I can make myself small again. Okay, so when we do mitosis, it's my arms, what? My legs, mitosis. It's for growth and repair. Okay, growth and repair. Meiosis is getting ready for sex. Okay, so how many divisions are we going to do? 2. Are we still going to replicate during the S stage? Yes, we are. Okay, here we go. So the whole meiosis getting ready for sex. The first stage is prophase 1. The chromosomes become visible. The nuclear envelope and the nucleolus disappear, radiating microtubules, asters, synapses, crossing over, which increases what? Variety. They meet in the middle at metaphase 1. And what are we demonstrating right there? Independent assortment. Big R can be with big T, or big R could be with, there we go. They meet in the middle at metaphase 1, away at anaphase 1, to nuclei at telophase 1, cytokinesis. We are now considered haploid. Why? The homologous pairs have been separated. The divorce has occurred. Prophase 2, metaphase 2. We did no crossing over. We have nobody to cross over with. We did no independent assortment. There's nobody to assort with, right? Metaphase 2, anaphase 2, telophase 2, cytokinesis, and I would have 1, 2, 3, 4 cells. If I was doing spermatogenesis, I would have made 4 sperm. If I was doing oogenesis, I would have made 1 egg and then 2 or 3 little polar bodies. Because we do unequal cytokinesis because we're the one who has to take that journey down the oviduct and supply all the nutrients. Okay, questions about that? Ask your bio buddy. How you doing on that? Sorry, delay of game. Okay. All right, so those are just the stages with descriptions. Okay, and then here you can see the rest. Now, cytokinesis, here you can see a cleavage furrow because that's an animal cell and they just kind of pinch off. But cytokinesis is different if you are a plant cell, because if you're a plant cell, you have to build like a cell plate and then a cell wall to divide your cells out because you don't pinch off like an animal cell. Animal cells don't have any cell walls. Now, bacteria, prokaryotic cells, do not undergo mitosis or meiosis because they have a single circular chromosome. They don't have linear chromosomes, and their form of reproduction is just asexual. Later I can tell you about sexual reproduction, but for the most part it is asexual. It's a clone of the parent cell, and it's just called binary fission. That one chromosome replicates and separates. Okay, we have already reviewed haploid versus diploid. Okay, diploid, you have 2 copies of information from both the mother and the father that made you, right? All of your cells are diploid, except whatever's in your eggs or your sperm. Those are the ones that are haploid. So when you made your sperm or your egg, you either gave your mom's chromosome 1 or your dad's chromosome, but you didn't give both. Foreshadowing to Mendel, what law is that? Law of segregation. Law of segregation. Is that making sense? Okay, so you're haploid because of the law of segregation. You only give one version. Then this right here goes through all the steps of meiosis 1 and meiosis 2, and I've already reviewed that with you. Can I move on past that? Okay. Then how does the process of meiosis increase variation, which is good for natural selection, right? And way 1 is crossing over. And when does crossing over occur? Prophase 1. Way 2 is independent assortment, which occurs when? Metaphase 1. And way 3 is because you made haploid cells, you need to rejoin them through the process of fertilization, and you don't know what sperm is going to fertilize what egg. And as I mentioned earlier, the formation of sperm, spermatogenesis, you'll make 4 haploid sperm. But when you do eggs, you do unequal cytokinesis. You don't divvy up the cytoplasm. You let one cell have everything and the other have just the nucleus, and that's it. And then you repeat that process again so that you have one large ovum, one large egg. And ladies, we do, bless you, meiosis 1 before we were ever born, and we don't do meiosis 2 until a sperm shows up. Right? And then here, non-disjunction is when you fail to separate, and that's when you can get trisomies and monosomies. And Down syndrome is a good example of trisomy 21, the most common autosomal trisomy. Then I gave you a slide here that you can use: mitosis, meiosis, differences, and then how they are the same. Okay, that is chapter 5. That's our oldest chapter of the unit, yes? Okay, so then this is chapter 22, okay? Again, don't forget your additional objectives. Can you name the structures of fertilization and then describe all the stages of embryonic development? Can you name and describe the layers of the gastrula and what each will become? Can you differentiate and give an example of induction and morphogenesis? Remember, induction is peer pressure, and morphogenesis is what that peer pressure gets, and that's movement of cells, right? Induction is the peer pressure. Can you describe how mitosis and meiosis contribute to the stages of fertilization and development? Meiosis will get you the haploid sperm and egg, but from there on out, what are you doing? Mitosis, doing mitosis. Okay, tell your bio buddy, what ones are you most nervous about of those that you need to really focus on? All right, so this first part is just showing you how the sperm fertilizes the egg. And remember, the sperm on its head, it's wearing a little beanie called the acrosome that has enzymes to digest the zona pellucida, the jelly coat of the egg. Then the sperm and the egg haploid cells fuse to form that diploid zygote. Remember, the sperm only contributes his nucleus. That's it. All the other, like the mitochondria, all of the other organelles come from your mother, right? And we can look at, do you remember that part? Okay. And then as far as the stages go, these are the different stages. And then why don't we go ahead and do that one as well while I'm thinking about it. Maybe. There we go. Am I in? Okay. So here it comes. Do it with me. Here it comes Mr. Happy Sperm. It is what? Haploid. The egg is haploid. They fuse and form a diploid zygote. It does cleavage, cleavage, cleavage. So it's just doing a whole lot of mitosis and kind of skipping through G1 and G2, but it's still replicating all the DNA and doing all of that, right? And that's why it's about the same size as the original fertilized egg. Okay, go again. Cleavage, 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 and you form a solid ball of cells called a morula. And then you're going to pump in salt. Who's going to follow? Water, because water must flow from the hypo. And you form a blastula. And a blastula is a hollow ball of cells. What do we call the cavity inside the blastula? A blastocele. Then we let out the gastrula, right? Let out the gas to form a gastrula. A gastrula is a layered ball of cells. We'll talk about those layers in just a minute. Back to your gastrula. And then you have the blastocele went bye-bye, right? And you have a new cavity inside. What's the new cavity called? Archenteron or archenteron. And what's the opening to that? It's called the blastopore, which becomes our anus. And then the last stage is the norula. And this is when you have that infolding. Little hint, know the stages and their order. Did you hear me? Know the stages and their order. I bet I will ask all of you that. Know the stages and their order and describe those stages to me. Okay? Now, come on now. There. Okay, good. Next, let's talk about the layered ball of cells, the gastrula. The outermost layer, we already know what it's going to be. Even in these early stages, we know it's going to be our what? Skin. I can tell I'm filling my skin because I'm using my nervous system and the lens of my eye. Okay, so what we're doing is we're just memorizing the ecto and endoderm because then everything else has to be the mesoderm. So tell me again, what does the ectoderm become? Skin, nervous system, and lens of eye. The endoderm, remember the archenteron, that inner cavity is your gut tube. So what systems can I access through my mouth here? Digestive and respiratory. Everything else, like musculoskeletal and all of those others, have to come from the mesoderm. Right? You okay with that? Okay. And then differentiation is that specialization. We know every cell contains the exact same DNA, yet all of our cells are so different and specialized. And that's because what's in the cytoplasm is different from the cleavage, cleavage, cleavage. When we were doing that, different ratios of chemicals were in each one of those, which sent them down different developmental pathways. So we know a couple things right off the bat. Where the sperm penetrates the egg, that's going to be your head or anterior region, and then the opposite of that will be your posterior. Wherever you have yolk, that's going to be the front, and no yolk is going to be the back. So even though it's just one cell, wherever the sperm hits that one cell sets in motion all of these different items, okay, in development. And that the changes that occur is called morphogenesis. The shape is morphology, is shape. Genesis is the beginning of that. And morphogens are these chemicals. These are the peer pressures. Remember how we talked about how the notochord tells the ectoderm above it to fold in and form the spinal cord? It's doing that with morphogens. That's how you apply the peer pressure. Those are the words to get it to happen. Okay, and then speaking of that, we can see it right here, where the notochord is telling the ectoderm to fold in to form the norula and form your spinal cord. And then later, the notochord, which is mesodermal tissue, becomes your what to protect that spinal cord? Your vertebrae. Okay, that becomes your vertebrae. And then we also know that death is really important in development too. So apoptosis is cell death used to create fingers or to create a difference between your radius and your ulna. Okay, those are examples of where that helps with the developmental process. Okay, that, and then here, our most recent chapter. Read through that. Can you name Mendel's 3 laws, describe them, and provide an example of each? Can you identify and differentiate between somatic cells? Let's do these right now as we go along. Want to do it as we go along, and then I'll show you the pictures? So what are Mendel's 3 laws? Law of dominance. Big T is dominant over little t. Law of segregation, meaning when I make gametes, I either send the big T or I send the little t. Law of independent assortment means I can make some gametes with a big T and a big R, and some gametes with a big T and a little r. Not a big T and a little t because of the law of segregation, right? I can make it with a little t and a big R and a little t and a little r. That's the law of independent assortment. I had a hand up over there. Could you go ahead? Hands now? Okay. Then, and I have slides to help you with this too, okay? Can you identify and differentiate between somatic cells and gametes? Somatic cells is just a name for what? Body cells. Gametes would be what? Not muscles. Yeah, sperm and eggs, right? I'm sorry, I was trying to make ovaries here and huevos rancheros. Okay, homozygous, heterozygous. Homozygous is when your 2 alleles are the same. It could be big T, or little t, little t. Heterozygous would be what? Big T, little t. When I'm talking about the T's, I'm talking about the genotype. When I'm saying tall or short, I'm talking about the phenotype. We good? Okay. Multiple alleles versus polygenic traits. Multiple alleles would be if I had big T1, big T2, big T3, and little t. Right? I don't just have big, little. Where did we see that? Blood types. Remember? We had big IA. What else? Big IB and little I. Is big I still dominant over little I? Yes. But we have options on our big I-ness. Yeah? Okay. That's multiple alleles. Polygenic is using I's, T's, R's, Q's, and P's to give you your trait like intelligence or skin color or height, right? We're not tall, short, we're all in between. We're not black, white, we're all in between. Okay? Codominance versus incomplete dominance. Codominance, again, back to the blood types. That's like AB blood or the roan cattle, right? Where it was neither just red or white. It wasn't just a blending. Blending would be more like incomplete. Red, white flower makes pink. Codominance would be red, white flower makes offspring that has some red petals and some white petals. Our rat, where all the hair appears gray, what would that be? Incomplete. Some of the hairs are black, some of the hairs are white. Codominance. Okay, nature versus nurture, right? I could have the genes to be really tall, but if I don't eat nutritious food, will I be tall? No. I could be a Himalayan rabbit where my body's supposed to be white, but if you put ice on my back, it will grow in black. Okay, that is where your environment is determining your phenotype. And you see this in like reptiles and sex determination. The temperature of the nest determines whether it'll be a male or female alligator. Remember Nemo? If you're Nemo and Nemo's population, it is better to be a female than a male. So when Nemo's mom, when she died, right, then the largest male would become the female. So that's in your environment. Okay. Nemo's dad could eventually be Nemo's mom. Follow? Okay. And then, can you analyze Punnett squares? Can we do Punnett squares? Yeah, we put gametes on the side and zygotes in the middle, right? And if we have to do a big cross, we do individual Punnets and we what? Multiply them together. Why would I do a test cross? Do you need to test me with my blue eyes? No, you know what I am, right? I have to be little b, little b. Who would we need to test? Because they have a dominant phenotype, but we don't know their genotype because they could be hiding it, right? Okay. And then recognizing the different pedigrees. What could you say for sure about dominant pedigrees? They do not skip generations, ever. Recessive, can they skip? Yes. Could two people who don't have it have a child who has it? Yes. Okay. Sex linked, you see it more in males than females. The other thing about sex link, if a girl has it, what can you tell me about her parents? If a girl has a sex link trait, do they both have to have it? Who has to have it of their parents? Their dad, because the dad has to give her a bad X. The mom could either have it or be a carrier. Can males be carriers? No, they cannot. They only have one shot at their X. They either have a good one or a bad one. Okay, so here for support, just so you know, this talks about the 3 laws. Say help if you need me to go over anything you see, okay? This, I want to remind you, these are your alleles. This would be what? Homozygous or heterozygous? And when you see this right here, is that a genotype or a phenotype? But if I say round, then it's the good. These genes are all on the same chromosome, so they are linked. They will travel together. Big S is traveling with little t, unless what occurs? Crossing over. Good. Okay. Then this is showing you a test cross, and you would expect if a brown-eyed person had 15 kids, they're very prolific, with 3 different wives. Just for fun. We'll move it, shake it up a little bit here. And all of the children happened to have brown eyes, and all of his wives had blue eyes, and all of his children have brown eyes. What do you know for sure? Yeah, he's homozygous. But if he has just one blue-eyed child, we know he was a heterozygote. Okay. We also looked at, this is your good example of multiple alleles, because we have big I A, big I B, and little I. AB blood is a good example of codominance, because both the A and the B are expressed. Incomplete dominance, a good example for that one is straight hair is H1H1, curly hair is H1H2. If these two produce a child, they will be H1H2 children. Their hair will be neither straight nor curly. It'll be wavy, right? And if this brother and sister decide to inbreed, I am not saying to do that. You would just have to know me. But if they decided to inbreed, what kind of children could they have? Troubled children. But what kind of hair? They could be, how many would you expect to be wavy? Half would be wavy, you would predict, and 1/4 straight and 1/4. But each time, could they just keep having wavy haired children? Yes, because chance has no. Good. Okay, on sex links crosses, remember, it's only the letters go on the X and only the X. Okay, there are Y genes, we're just not dealing with them. And all of the sex link traits that I give you will always be what? Not dominant, but recessive. Okay, for girls to have it, they have to get 2 bad ones. Are any of the girls going to have it here? No. These would be called what? Carriers. What's this one? And this one. They have it, right? Okay. And then polygenic traits. This is where you see that bell-shaped curve. Instead of like tall, short, you see all the intermediates in between. That's typical of polygenic traits. Here is where you see the environment influencing the phenotype. And then this is showing you linked genes, right? And the only thing that breaks up the linkage is crossing over. And then this was just kind of the cues for when you're looking at pedigrees. And then the last slide here says, this exam is more about memorizing. It is understanding, because I'll just ask you a series of questions to have you apply it. Okay? Good job, you guys. I'm proud of you. Yes, hang on one second. Let me just